Welcome everyone. On behalf of the Statistical Society of Australia, I would like to welcome you to this webinar. My name is Sabine Braat and I work as a biostatistician at the University of Melbourne. And I'm the co-chair of the Society's Biostatistics and Bioinformatics section. During the next hour, we will learn a bit more about one of the great challenges with single-cell RNA sequencing data related to the so-called dropout phenomenon in a presentation by Associate Professor Agus Salim. So Agus works at the Department of Mathematics and Statistics at La Trobe University in Melbourne, and he came to La Trobe in April 2013, having previously held positions at the Australian National University and National University of Singapore. Agus has over 15 years experience as a biostatistician and has extensive records of collaborations with clinicians and epidemiologists. He has received competitive funding from the NHMRC for his applied and methodological work, and his main methodological interest is in the area of survival analysis, with a focus on analysis of nested case control data and the analysis of high throughput data from next generation sequencing. So before handing over to Agus for his presentation, I have one practical announcement. So the webinar will last about an hour and we will allow for questions. I would like to ask each of you to please save your questions until the end of the presentation. And if you have any question during the presentation, you are most welcome to let us know, but use the chat log for that, and we will aim to address the question at the end of the presentation. So without further ado, Agus, please feel free to presentation whenever you're ready. Thanks.
Thanks, Sabine, for the introduction, and I would like to thank the Biostatistics and Bioinformatics sections of the SSAI for giving me this opportunity to talk about my research. So the title of my talk today is Modeling Molecular Dropout in Single-Cell RNA-seq Experiment that leads to improved identification of marker genes. I think this is a very, this era is a very exciting era for us statisticians, because, you know, day by day you're seeing with the kind of advance in technology, and not only you know, in biology but in other fields as well, you see a new type of data coming in every day, and it's certainly a challenge for us to kind of develop methods that can cater or can accommodate the features of this new data. So this is the outline of my talk today. So I'm going to briefly introduce you to a single-cell RNA-seq technology and a little bit of the typical data analysis. Then I'm going to talk about how I address some of the challenges when analyzing this data. In particular, I'm going to talk about our approach called DESCENT, and that stands for Differential Expression for with capture efficiency adjustment to modeling molecular dropout in this kind of experiment, followed by some empirical evaluation, and then an example of application, and I finish with some key points. Okay, so as a background, in the last few years, okay, with a technological advance has enabled us to perform sequencing at a single cell level. So those of you who are familiar with, say, next generation sequencing, where you perform sequencing at the bulk level, and that is you're sequencing 1,000 or 10,000 of cells together, okay? Now we are able to do this for each cell separately. And this has enabled discoveries of novel cell types and better understanding of cell heterogeneity and differentiation. Right? And certainly this is a hot topic. I mean, if you see this is a bar chart of number of publications in the last few years up to 2015. You see there's an exponential increase in the publication in PubMed that they use single cell data. Okay, so the technology itself, because we are going to perform a sequencing for each cell separately. So one of the first key step is to sort the cells. And in these aspects, there are a lot of technology to do this. And I'm just going to give two examples here. You can use, say, the Fluidime machine here, where, you know, the machine will actually sort the cells and put them into plates. Okay, it's either 96 plate or 384 plates. So each cell will go to each well, right? Or you can use a droplet-based technology. So things like a 10x machine actually use a droplet-based technology where you kind of put your cell through this apparatus here where the cell will be encapsulated by a kind of oil solution, so forming a droplet, and that's how you separate the cell. Once the so a typical workflow, okay, for this kind of data. First of all, you have to bear in mind that we are dealing with cells where the starting material is very, very low, right? So we already have a problem with very low kind of a biological material. And you have a cell-to-cell variation in size because, you know, we're talking about when you're sorting the cells, you don't actually know the cell types, okay? You don't know the cell type identity. So think about blood samples, that there will be T cells there will be B cells, but we actually don't know the identity of the cell type, right? And there are cell-to-cell, there are variation in a cell-to-cell size that we need to be mindful about. For example, if you're comparing, you know, one cell to another cell with a bigger volume tends to have higher number of molecules, of course, and that needs to be kind of normalized before you can perform further analysis. Otherwise, you know, you'll be looking at differences that may not be meaningful biologically just due to the cell size volume. There's also issue with low molecule capture rates, and this is what I'm going to concentrate later. Right? The workflow itself, as I said, you start with isolating the cells, and then of course you extract the RNA, you know, through reverse and then through reverse transcription process, you're turning the RNA into cDNA. Okay, and this is where you can actually add what we call UMI. So each molecule can actually be tagged using a UMI barcode. Okay, so at the end, this will enable you to what you will have at the end after, you know, the amplification process, okay, and then you perform library preparation and then performing your sequencing. At the end, what you will have is a matrix, okay, of what we call a UMI count. So this is a basically matrix of number of molecules, where typically the column is a cell, so each column is a cell, and then the row they are genes, right? There's also a feature called spike-in. So as a measure of quality controls, you can actually add spike-ins to each cell, and typically spike-in is added before the transcription process. So when before you turn the RNA into cDNA here, and the most popular spike-ins that has been used quite popularly is called ERCC. That's stand for External RNA Control Consortium. This is a 92 kind of artificial transcript, okay? And each have their own kind of a non-concentration that you actually add or inject to the cells, okay? So these are transcript where you know approximately the quantity, the number of molecules. Okay, the typical analysis itself. Now, first of all, quality controls, I mean, I cannot emphasize enough that when dealing with single-cell RNA-seq data, quality control is very important. So you need to remove differences between cells that are just due to, say, cell volume. This is achieved through normalization, for example. Okay. These differences is due to cell volume and capture rates. And also you need to remove cells that are kind of low quality. So often we use the kind of the amount or the percentage of molecules that come from mitochondrial, for example, as a marker of low quality cells. So cells with high content of with high mitochondrial contents are often marked as low quality cells that need to be removed from further analysis. But assuming that you have done your quality controls correctly, right? Typically, you would do dimensional try to cluster the data. Okay? So you're clustering the data, like in this example here, you have 10 clusters, and the hope is that each cluster would correspond to more homogeneous cells, cell with more homogeneous characteristics, like say each cluster correspond to say one cell type. And this is very important because remember when we are performing the cell sorting, we do not actually know the identity, the cell type identity of those cells. So performing this cluster analysis is very important because often you want to, once you identify the cell types, what you want is you want to identify the marker genes, for example, for each cell type. What are the marker genes for certain cell type, for example, and that is achieved through differential expression. So this is typically involving comparing two or more clusters, okay, and looking at genes that are differentially expressed between those clusters. And this is what I'm going to concentrate today on, the differential expression analysis. There are also other analysis like Pseudotime. We call it a Pseudotime analysis. So what you do is you use the UMAP or the PCA representation of the data and use a minimum spanning tree to order the cells, okay, along a tree. And again, the hope is that when you're ordering the cells along a tree like this, then you know, the start and the beginning of the tree may correspond to, say, early development phase of a cell, and or late development late development phase of the cell, right? And that gives you an idea about, you know, which cells are at different kind of phase of development. All right. So I mentioned about the low biological materials in each cell and the low capture rates, right? And these two factors actually contribute to a feature in the well-known feature in the single-cell RNA-seq data, where up to 90% of the count matrix actually consists of zero. So often you don't actually see any molecules at all for a lot of the genes, right? And these are the 4 examples data set that I'm going to use to test our model, DECENT. And you can see here that they come from different sequencing protocols. Okay, some have spike-ins and some don't. What you can see here is that the proportion of zeros varies, right? But sometimes it can go up to 90%, right? And this, as I will show you later, this 90% of zeros doesn't mean that the genes are silent. It doesn't mean that there is no molecule in that gene, but it's rather that the molecule has not been captured by the technology. Okay. Okay. As an evidence of this low capture rates, I'm going to give you an example here where we have the spike-in data. So these are 1, 2, 3. These are the a particular cell, where you see on the x-axis here is the nominal number of molecules that has been added, because remember, this is transcript. We know how much we put in. And these are the number of molecules that are observed. And what you can see is that it's always that we observe less than what we added. So not all the molecules that we added has been captured, right? And if you look at the capture rate, so if you calculate the ratio of how many you have added, how many molecules you have added, and how many that has been captured per cell, you see that there are variation, but it is not uncommon to see less than 10% of the molecule actually has been captured. Okay, this is still with transcript again, with spike-in transcript. Here you can see that on the y-axis here, that's the proportion of cell with 0 count for that transcript, right? And This is on the x-axis, this is the log concentration. As you can see, there's a very nice curve here. This is the dropout curve that we fit. And what you can see is that for higher abundant transcript, okay, you have less of a zero count, right? And for lower abundant transcript, we have a lot of zero count here. And we call this zero count here technical zeros. Well, why do we call it technical zeros? That's because the zero count happened due to the imperfect technology. Okay, so it's due to technical factors rather than biological factors. Right? There are other type of zero count. I will introduce that later, and that's called biological zeros. But these are evidence of technological zeros. All of this, where you have almost 100% zero count, those are technical zeros. Okay. So this imperfect capture result in total, okay, of molecule dropout where you don't observe anything, or it can result in partial dropout in the sense that you still observe something, but what you observe is actually less than what it was. Okay. Now this is for endogenous genes. So endogenous genes are genes that are part of the sample that of interest. Okay, say if you have a human sample, say these are human genes. Right. So again, you can see that very similar curve there that you observe in the spike in transcript for the endogenous genes, right? In fact, if you fit that curve, it will go to the middle of the blue points here. What you can also see is that for some genes, okay, like this one, for example, the proportion of zero count is actually much more than what would be predicted if all the zeros are just technical zeros. So this is actually an evidence that there are biological zeros as well in the data, and these biological zeros are the zero count that are due to the gene being silent, right? So we have two types of zero here: the biological and the technical zero. Okay. So just to kind of recap, we have two kind of zero. Okay, so if you observe zero, it can be because the gene is silent. Okay, in which case it doesn't matter what how you capture it, you ended up with zero count, or the gene is actually expressed but the molecules are not captured, right? And you ended up with technical zero. And we are going to try to model this dropout process. Okay. Another feature of the single-cell RNA-seq data, and in fact this is shared with bulk data as well, is that the data is over-dispersed. So here the red line is the log CV square, that's the log of coefficient of variation square as a function of abundance, and that's what predicted if the data follow kind of a Poisson distribution. As you can see, a lot of the points actually above the line, which suggests that there are extra Poisson variation in the data. Right. So just to recap that observe single-cell RNA-seq data. First, they are over-dispersed, okay, and they contain a lot of zero count, okay, and most of these zero counts are technical zeros, but some are or some of which are maybe biological zeros as well. So we need to take into account this biological zero in addition to the over-dispersion. Right. So how am I going to kind of model the dropout event? We started with a very simple kind of model. Well, so here, let's say you have Yij, which is the unobserved number of molecule for spike in I that has been added to molecule, added to cell J, that should be cell rather than molecule, and Zij is what is actually observed. So remember, Yij is things that you do not observe, okay? You do not exactly observe, but Zij is things that you actually observe, okay? So even for spike-ins, you don't actually exactly observe what Yij, although you know approximately how much it is, okay? You know the average molecule count that has been added for transcript, but you don't actually know exactly what it is. So here, we will play it as simple, and we will assume, say, because it's count data, we are going to assume that the unobserved number of molecules for the spike in I actually follow Poisson with a known concentrations, right? And then followed by that, we have what we call a dropout model, where given the number of unobserved number of molecules, the observed number of molecules is a simple binomial sampling, right? So you can imagine you have k molecules in the beginning, then you're trying to capture this molecule where the probability of capturing each molecule is cell specific and given by eta j here. The nice thing about this is that this specification leads into a kind of cross-form formula for the marginal probability of the observed data. Okay, so Zij is another Poisson basically, where the eta j comes into the equation as a multiplicative factor there. So we have 4 plus 3, okay, data sets. So we have 7 data sets here, 4 plate-based and 3 droplet-based. They come from a variety of technology where we have the spike-ins for this data sets. And why we start with spike-in, because remember with spike-in we know the approximate concentration, so that's making it kind of easy to concentrate on modeling the dropout without having to estimate the unobserved concentration. So what we see though is that this is for one of the data set. Okay, so you would expect that if your dropout model is good, okay, so when you calculate the chi-square goodness of fit statistics, okay, for each cell, and then you have the histogram or the density plot here across all cells, you would expect that the observed, okay, density to follow chi-square, right, with certain degree of freedom. Yeah, and that is given by the gray curve there. But look at here, you can see that it's actually quite off, right? So the binomial dropout model actually does not fit the data very well. And this is where we actually started looking at a different dropout model. And why the binomial dropout model kind of failed, despite the fact of the nice thing of the marginal probability for the observed data kind of giving you a cross-form formula, so that's very handy in terms of computational, but it has failed. And one of the reasons is because what happened with the binomial dropout model is you're assuming the same capture rates for all the genes, right? Remember, if I go back here, you're assuming the same capture rate for, so capture rate is cell specific, but within a cell, the capture rate is actually the same for all the genes. So that might not be realistic, right? So we then develop what we call a beta-binomial dropout model. So with the beta-binomial dropout model, we, instead of fixing the capture rates, we place a beta prior on the capture rate. So we are allowing uncertainty with the capture rate. So now each gene within a cell have their own capture rate that follow a beta distribution. The only restriction that we make is that the average of the beta distribution needs to be equal to the cell level capture rates within a cell, right? And the variation in the beta distribution, okay, the uncertainty around the mean is controlled by this parameter here. We call it rho, okay? Now, the drawback with this is that there is no cross-form formula for the observed data, right? So we have to do a lot of integration in order to perform estimation and inference. But let's forget about that for now. Let's forget about the difficulty in computation. So this is the beta distribution that I was talking about. So with beta distribution, you have the mean, okay, and then there's uncertainty around that mean, and the uncertainty is actually a function of the A and the B, okay. So if A and B going to infinity, for example, then you have less uncertainty, so rho become very small. So when rho is small, that means there's less uncertainty about capture rate for that particular gene around the cell level mean. But when rho is big, then you know, you have more of uncertainty. Right? So now we are going to compare the beta binomial versus the binomial dropout model. So here is from this is from one dataset, the dataset that I mentioned in the beginning. What you can see is that the beta binomial actually provide a much better fit to the data. Okay, and if you look at across other datasets, now beta binomial, the blue one, does not always provide kind of the best fit or the satisfactory fit, but in each case, it's actually much better than binomial. Okay, so beta binomial is always perfect, but if you compare it to binomial, it's much better. That's so that's one key point here. That's why we decided to use a beta binomial dropout for our model. Second thing is that when we so we play around with the rho parameter here. So we are allowing the rho parameter to be this is for spike-ins. We are allowing it to be kind of different for each spike-in. What you can see is that the rho. So here on the y-axis, that's the logit of the rho, and then on the x-axis is really the log abundance for the transcript. And what you can see here is actually there is a quite a nice linear relationship between that parameter, which is the logit of rho, and the abundance of the transcript. And we are also going to take advantage of this in our model. All right. So this is our model. Okay, this we call it decent. It's a differential expression with capture efficiency adjustment. So first we have the unobserved pre-dropout count. Okay, and we use a zero inflated negative binomial where there is a phi naught parameter here. That's it. That is gene specific, and that actually controls the amount of biological zeros. Remember, we have shown that in our data, sometimes the gene have biological zeros, right? And this is the mean parameter, where for each cell j, there is a multiplicative factor here, Sj, and that is actually representing the differences in the cell volume. Remember, I said that cell with different volume may have different total number of molecules. So that is scattered through the S parameter there, Sj parameter, and that is just a dispersion parameter for the negative binomial. Okay, so this for the unobserved data for Y. So then we have a dropout model, and which is, you know, a binomial where, with a beta prior on the capture efficiency, and on the capture rates parameter, okay, which gives you overall a beta binomial dropout model. Okay, now because we are interested in primarily interested in performing differential expression, that is comparing two different cell types, for example. We cater for this differential expression analysis in our model. So the mean parameter mu can be made as a function of x, where x is, if you have two cell type, it's just a dummy variable, it's 0 or 1. And mu can also depend on other variable, w here. So the w is where you can put things like, you know, the batch identity, if you believe there is a batch of x, or things like cell cycle phase, if you think that the gene expression is actually affected by a cell cycle. And as I mentioned before, for the beta prior, we are restricting this ratio here between A and the sum of A plus B to be equal to the cell level capture rates. Okay, and this cell level capture rate is actually estimated from the spike-ins. And for the logit of the rho parameter, okay, that is modeled as a kind of a linear function of the abundance. Now you must be asking why we are using zero inflated negative binomial for the unobserved data. Where do we get the evidence from? Well, we get the evidence from this, what we call single molecule fish data. So a single molecule, I think it's fluorescence in situ hybridization data, where you can actually observe the number of molecules in each cell directly without having to perform sequencing. Okay, so in this kind of technology, it's very expensive, but there is no dropout because you can observe everything in the data. And what we observe is that here, panel A here above, it's where we test for over-dispersion. So we are comparing a fitting a model without over-dispersion parameter and model with over-dispersion parameter to the SM fish data. And what you observe is that if the over-dispersion parameter is not really needed, you will see that all of this point, okay, will line along this line here. That's the expected minus log 10 of the p-value, and that's what is observed. But what you can see here is that for all of the genes, the over-dispersion is actually needed. Okay, lower panel here, that's when we compare model, where we compare model with and without the zero inflation parameter, the pi naught. Remember, that is the one that's controlling the amount of biological zero. So if you don't have if you're fitting a model without pi naught, then that's amount to believing that there is no biological zero in your unobserved data. And what you can see here is that for a minority of the gene, the biological the parameter associated by biological zeros actually needed. We found about 5 to 6% of the genes actually need a biological zero parameter, the zero inflation parameters, and that actually quite in concordance with what we see in other data sets as well. So for the majority of the genes, these parameters are probably not needed, but there are 5 or 6% of genes where the unobserved number of molecules actually have biological zeros. Okay, so we come to the estimation, right? So we have the model here for the unobserved data. We have the model for the dropout. How do we estimate the parameters? So here, what we are going to do is we are going to treat this as a missing data problem. So we will treat the complete data being the Y and the Z. Okay, of course, we only observe Z. Okay, that is our observed data, and our complete data is Y and Z. And the parameters that we need to estimate here, okay, bear in mind that you have the cell level parameters, and these are the taus that are associated with the dropout model, and you have the S here, that is the parameters associated with modeling the differences in the cell volume, and then you have the gene level parameters here, okay, for each gene that's associated with the zero inflated negative binomial specification of the unobserved data. One thing that I want to say here is that we are not estimating the capture rates. Okay, the capture rate is assumed to be known for each cell, and we use Spikin to estimate this. Okay, so if you look at the log likelihood of the complete data, okay, some of you who are more kind of theoretically oriented may want to look at this. So the log likelihood of the complete data, because the complete data is Y and Z, consists of two parts. The first part here that comes from the zero inflated negative binomial specification of the unobserved data, and that comes from the beta-binomial specification of the dropout model. Okay, so that's you can see here that's the first component, and that's the second component. One thing that you can see here is that in EM algorithm, eventually when you're performing estimation, you need to take the expected value of the log likelihood of the complete data. And you can probably see here that you can imagine that when you log this, you probably then need to take the expected value of this quantity here and the expected value of this quantity here, which is the unobserved data. And you are right. So when we're performing the E step, okay, two things that need to be evaluated in the E step in terms of the expected value is if the gene have 0 count, okay, what is observed is actually 0, we need to estimate the probability of the technical 0. Okay, so the probability that Y was actually not 0, but what we observe is 0. And for all entries, okay, we need to evaluate the expected value of the unobserved number of molecules given what we actually observe. Now, because the beta-binomial dropout does not lead into closed form formula, evaluating this two expected values actually require kind of summation. So what we do is computationally, we approximate the summation using integral with changing in the bound to reflect the continuity correction, and then we evaluate the integral further using a Gaussian quadrature. Okay, so with the EM algorithm, you have E step and you have M step. In the M step, what we do is we first replace this quantity here in the log likelihood of the complete data with this quantity that has the probability of technical 0 from the E step, and the unobserved data is replaced by its imputed version here again from the E step, and then given that, then we maximize the likelihood of the complete data numerically, right? So here what we do, this is just, I'm just going to explain very briefly. First, you know, of course, you need to have initial parameter estimates. You use the initial parameter estimate, and then given the gene level, sorry, the cell level parameters, you update the gene level parameters, okay, numerically, and then given the cell level parameters, then you update the, sorry, given the gene level parameters, you then update the cell level parameters, and you do that consecutively. I'm not going to go through the detail here until you achieve convergence, but of course, that is the estimation, but we cannot use the imputed values to perform inference directly, right? Because those are the imputed values from EM algorithm, and if you use that directly, you don't take into account the uncertainty in the imputed values. So for inference, we are actually using the log likelihood of the incomplete data. Okay, and that's the marginal likelihood for Z, and you can see it's full of some horrible summation. And again, computationally, when we do this, we have to approximate this using Gaussian quadrature. Okay, so that's the theory part, right? So, but we then need to see how this model actually performs. We perform first a simulation study where we compare our method against the others. So this is for 2,500 genes. We let about 10% of the genes to be differentially expressed. So there are 2 cell types. Okay, the simulation parameters actually estimated from a real data set. Okay, and that is from a data set called Tung dataset. That is part of what we are going to compare. As you can see here, in terms of estimating the parameters back, okay, the mean parameters is very good between true and what is estimated. Zero inflation, well, it's okay. Okay, it's a bit hard estimating the inflation parameter, but you can see there's no bias basically, and also the dispersion parameter. So we are quite happy, I mean, with that. And then when you look at the how our method actually perform, okay, compared to other methods, because there are a lot of other methods for performing differential expression involving single cell RNA-seq data. There is MAS, SCDE, monocle, even EDGAR. People use EDGAR. EDGAR was developed for about data, but people also use it sometimes for single cell data. And on the simulated data, you can see that this is the ROC curve. Okay, you can see that our ROC curve is always kind of the highest, but that's, you know, that's actually Not very surprising because we simulated the data under our model, and of course, our method should come out the best in this regard in terms of giving you the best ROC and the smallest kind of false discovery rates. Okay, but then we will see how our data actually perform with real data. Okay, and if you look at the kind of the ability to estimate the proportion of biological zeros. Okay, remember this is simulated data, so we know for each gene here, each dot point is a gene. We know what is the proportion of the true proportion of biological zero, and that is what is estimated. And again, the true number of molecules before dropout. Okay, and that is what is estimated after our EM algorithm, and you can see there's actually a good concordance there. Okay, and one thing that I will also mention is that if you use DSEN to estimate the data, to simulate the data. Okay, so you have a real data set, you estimate the parameters, and use DSEN to simulate the data. You see that the simulated data is actually very similar to a real data set. So that's real data set in terms of relationship between abundance on the x-axis and biological coefficient of variation. You can see that ours is actually not too bad, I mean, compared to the real data. Okay, and this is actually Splatter. Splatter is quite popular for simulating single-cell RNA-seq data, but yes, as you can see, ours is doing a bit better there. This one is doing a bit better. Okay, so these are the real data set. Okay, so in a way, this is the real challenge. You apply your method into a real data set and see how it performs compared to others. So for this real data set, we have gold standard DE genes. So we know which genes are going to be differentially expressed. Where do we get this from? We get this from bulk samples. So we have matching bulk sample for this 4 data sets. So and we derive the gold standard DE genes from bulk samples. And use that to construct the ROC curve. And you can see DSEN always come out on top, okay, the blue line there in terms of the ROC curve. I mean, sometimes the advantage is not that great, okay. For Somilon data, the advantage is actually quite good. But for other data sets, it's not that much. Although the second best method is not always consistent, okay. So after DSEN, the other method is not always consistent in terms of their ranking in the performance. Okay, this is the FDR. I'm going to skip this. Again, it conveys a message that DSEN actually give you the smallest false discovery rates. In terms of controlling a type 1 error, okay, so you have one of the data set where we actually take one particular cell type and just split them into 2 and perform a kind of a null experiment, because when you're comparing the same cell type, you expect all the genes to be not differentially expressed. So anything that come out differentially expressed, that's actually a type 1 error. Okay, and as you can see, in terms of the distribution of the p-values under the null experiment, DSEN is very good. It's almost close to a uniform distribution, because that's what expected under the uniform distribution. Some of the other methods are not as good. Okay, so we try to look at or try to understand better what actually drive the DSEN performance here. Why sometimes DSEN actually perform, the performance gains is much kind of better compared to other methods, and sometimes it's not. One thing that we notice is that these are the logit row parameters. Remember, that's actually controlling the amount of uncertainty with the capture efficiency or the capture rates in the binomial prior. So if rho is high, that means there are a lot of uncertainty in the beta distribution, okay, around the mean, and that actually leads into an over dispersed dropout model compared to binomial. As you can see, with Somilon data, where we actually confer the highest advantage, the logit row parameter is actually the largest, which actually tells you that decent benefit is actually more apparent when the dropout is more of a disperse. Okay, so decent essentially use imputed data, so performing inference indirectly on the imputed data. And as you can see here, these are some genes for one of the dataset, where we know that these are gold standard DE gene, these genes are differentially expressed. So you can see that what happened is the violin plot before imputation, okay, and that's the violin plot after decent imputation. You can see that after decent imputation, there's more of a gradation between the two cell types, okay. Again, this is showing the advantage of working with the imputed data rather than the unimputed one. If you look at the correlation, okay, this is for unimputed data, okay. And that's for the imputed data. As you can see, these are the two genes where we expect the genes to be kind of positively correlated. Again, if you work with the imputed data, you will see that the correlation is much more obvious. So in a way, the imputed data actually increase the signal to noise ratio. And in the bottom panel here, you see that these are two genes that should be negatively correlated. And with the imputed data, unimputed data is not very obvious, but with the imputed data, it's much more obvious that they're actually negatively correlated. Right? So just going to, this is the last section before conclusion. So I'm going to show you an example of DESCENT. So we use DESCENT to identify marker genes in this project called the Tissue Resident Memory Project. So this involves triple negative breast cancer patient, right? So as you know, T cells are involved in the kind of immune response, okay? Some of the T cells, once it encountered antigen, okay, it actually preserves memory about those antigen, and it remains in the tissue, okay, rather than in the blood circulation. And we call this tissue resident memory T cells, and they has been considered as an important target for immunotherapy. Okay, so we have two data sets here, come from two triple negative breast cancer patients, where they've been sorted using facts based on CD3. So you know these are T cells. So these are the clusters. Okay, these are the, so when we cluster the data after QC, we get these clusters here. We get 10 clusters. Okay, and our interest is actually the clusters corresponding to CD8 TRM. So CD8, so the tissue resident memory T cells that are expressing CD8 markers. Okay, and we, in our case, that's actually the number 2 here, number 2 clusters. In order to, we use DESCENT to derive the marker genes of this of each cluster, comparing it to other clusters, and well, according to our collaborators, the biologists, we come up with these marker genes for these clusters, and these are obviously the CD8 TRM clusters, number 2, and we so DESCENT managed to identify these marker genes, okay, and we are not satisfied there, so we also compared to our result, okay, to the bulk data. So we have the matching bulk data here. So we don't only have the single cell data, we have the bulk data as well for the same patients, and for it's also for CD8 T cells. So we are comparing here the TRM, the TRM T cells, the CD8 positive TRM T cell with the CD8 positive non-TRM T cells. As you can see here, these are the volcano plot for the comparing the CD8 TRM versus CD8 non-TRM T cells from our single cell data. These are the kind of the marker genes that are differentially expressed. So these are showing kind of marker of TRM activities. Okay, and this is what you get from the bulk data. So this is from single cell data. Okay, where there are a lot of dropouts, we have to perform inference with a lot of dropout, and this is from the bulk data. right? When there's not much of a dropout, and when we perform enrichment analysis, the list of differentially expressed genes here from the single cell actually contain a kind of high degree of concordance, or they are quite kind of concordance with what you observe in the bulk. Okay, so this is another evidence again that we managed to kind of extract information from data that are much lower quality, the single cell, and then what we get is quite similar to the result that you get from the bulk data. Okay, and when you compare this, when we use another data set, this is population level data set, and we use this marker to compute the gene score between CD8 TRM and non-CD8 TRM. You can see the quite a nice separation. So people with a higher kind of CD8 TRM activity actually have a better survival compared to those who do not. Okay, so these are some key points. We have developed DESEN. This is a DE method that adjusts for a dropout phenomenon due to imperfect molecular captures. And then this is developed by using empirical evidence, okay? And the performance gain over other methods is especially strong when the dropout process is over dispersed. Our future works, in the near future, we are going to try to improve the scalability of our methods. At the moment, it's working okay on the HPC, but we want to make this work also on the laptop. So we will do further speed up. And then we need to perform extension of this method to non-UMI data, because some single cell data actually does not have UMI tag. Okay. And then handling of batch effects and other general unwanted variation. I'm also recruiting, so interested PhD student can email me at this email address here. And these are the papers. DESEN papers have been published in bioinformatics. Okay, DESEN is implemented as an R package, and these are the TRM papers for which we are contributing authors using DESEN to identify the marker genes. I'd like to thank my collaborators at WIHI, Peter, Mark, and the Baker. And also like to acknowledge Matt and Louis at WIHI that have introduced me to this single cell world. Thank you very much.